Good afternoon, everyone. Thank you for joining us. We're in the backyard. I'm Steve.
And I'm Scott.
And together we're Backyard Music Science and Technology Channel.
Yep. And today we're talking about women and pain.
Yes, they have
So-
a great higher threshold. I don't know. Hormone-regulated immune cells produce IL-10 to resolve pain more effectively in males, offering a potential new target for non-opioid chronic pain treatments. Chronic pain often lingers longer in women than in men. A new study suggests that differences in immune cells, known as monocytes, we talked about that before, could be a key reason. These cells are regulated in part by sex hormones. Another binary, man or woman. Yeah. Would you like to know more?
In research published in Science and Immunology on February 20th of this year, scientists at Michigan State University identified a specific group of monocytes that release a pain-reducing molecule. The team found that these cells are more active in males, likely because of higher levels of sex hormones such as testosterone.
Okay. In females, monocytes produce lower levels of this pain dampening signal, which was linked to prolonged pain and slower recovery. Jeffrey Laumet, MSU Associate Professor of Physiology, and Jaewon Sim, a former graduate student in his lab, observed the same pattern in both mouse models and human patients.
The work, supported by National Institutes of Health and the Department of War, that's interesting, suggests it may eventually be possible to encourage these immune cells to generate stronger pain calming signals. Although practical treatments are likely many years away, Lamet believes that findings could ultimately lead to non-opioid options that provide relief for millions of people, while also reinforcing that women's pain has a clear biological basis.
Yeah, I can see how Department of War would be in on this to try and control the pain for soldiers and stuff, medical reasons.
Okay.
Pain begins when neurons throughout the body respond to stimulation. Under normal conditions, these nerve cells remain quiet until triggered by an injury such as a stubbing, a toe, or falling off a bike. In people with chronic pain, however, these sensors can become active with very mild stimulation or even without any obvious trigger.
Clinicians still depend heavily on self-reported pain scores, usually asking patients to rate discomfort on a scale from 1 to 10.
Every time you walk in, they ask you that.
Yeah, and that's a tough deal. I mean,
subjective,
I guess. Because pain is highly personal and subjective, differences in reporting can complicate diagnosis. When women report chronic or long-lasting pain more often than men, those differences are sometimes attributed to perception rather than biology.
Oh yeah, that's how the woman perceives it.
They're probably remembering the fight that they had with their husband the night before or something
or
like that.
Or the dream they had about their husband pissing them off, and then they wake up and it's real. You know what I'm saying?
Like I just said,
you know what
the
I'm
fight
saying.
that they had the night before.
Laumet has focused his laboratory on understanding pain for the past 6 years. During a small pilot study, his team noticed that males showed higher levels of interleukin-10 or IL-10. A second round of testing confirmed elevated levels of this molecule, which signals neurons to quiet pain responses, strengthening their confidence that the pattern was meaningful.
To investigate further, the researchers used high-dimensional spectral flow cytometry.
That sounds impressive.
Okay. SFC?
Yeah.
An advanced method for analyzing immune cells. They discovered that monocytes, once considered mainly precursor cells with limited direct function, actively communicate with pain-sensing neurons by releasing IL-10. The team found that IL-10 producing monocytes were significantly more active in males than females. When male sex hormones were blocked, this effect reversed.
Cause and effect right there.
Okay.
The researchers conducted at least 5 different types of experiments in mouse models to confirm their observations. Each approach produced consistent results. Laumet then collaborated with Sarah Lindstedt of the University of North Carolina at Chapel Hill, who studies psychological outcomes in people involved in car accidents.
Okay.
Her data revealed a similar trend. Men had more active IL-10 producing monocytes, and recover from pain more quickly.
Together, these findings highlight an immune neural pathway that influences how pain subsides, shifting attention from the triggers of pain to the mechanisms that allow it to persist. Future research will explore whether treatments can safely enhance IL-10 production along this pathway. Instead of simply blocking pain signals, such as therapies, could help the body resolve more pain more effectively.
Quote, future researchers can build on this work, Lamet said. This opens new avenues for non-opioid therapies, that the Army wants that, Marines, aimed at preventing chronic pain before it's established.
Well, opioids, man, they can take you over, right?
Yeah. Turn you into
quick addiction.
Yeah.
Quick addiction.
Make you steal in order to get more opioids.
Yeah, we don't want that.
Not that I know anything about that.
Right.
All right,
All right.
folks.
See you tomorrow.
We hope you enjoy your Sabbath.
Yep.
We'll see you on
Have a great
Monday.
Sunday, and we'll see you on Monday. Thanks.